Okay, thanks for the opportunity to talk a little bit about how we can harness the power of metagenomics to build the mine of tomorrow. And I want to reference that term because it actually comes from one of my colleagues who'll be presenting today, Scott Dunbar. And here's a quote from Scott. "Mining's flight to scale is an astonishing human achievement. Some of the world's largest mines have 500,000 tons of rock every day and can be seen from space. But scale has not solved the fundamental problem in mining. As ore bodies become more difficult to find and to mine, productivity declines." And so what we're really interested in conceiving of is a situation where we can harness the power of biotechnology to, in some ways, transform current mining paradigms into more of a bio refining or a biofactory context. Remineralizing or mineralizing rock masses and scrap metal and mine tailings, sort of waste to value processes so that we can actually recover metals, produce materials and products that have economic value. And what we're going to do today is explore how microbial communities, collectively termed the microcosmos, can be harnessed to realize some of these ideas. We're going to go through the looking glass of metagenomics, specifically how we study microbial communities using high throughput sequencing methods and computational pipelines to interpret that information in a mining context. And then we'll talk a little bit about the concept of a biomining knowledge base that could be used by both academics and practitioners alike to develop a community of practice around microbial biotechnologies in mining. So we live in a world that is dominated by microorganisms. Prokaryotic microorganisms, in particular bacteria and archaea, account for approximately 10 to the 30 cells. This is a very large number. It's more than the number of stars in the known universe. It's more than the collective synapses of all of humanity combined. And it's that power of large numbers that really gives microorganisms their impact on planetary scale processes. This is a wiring diagram of planet Earth metabolism, if you will, the way that reactions connect at planetary scales. And microorganisms mediate all of these processes. So they are the engines that drive Earth's biogeochemical processes and have been transforming the Earth over billions of years of cellular evolution. And they do that through networked metabolism. So this is a network. It could be a social network, a banking network. In this case, it's a microbial network where the links between nodes, which represent microorganisms, the links represent connections. They represent some kind of metabolic association or co-occurrence pattern. And what we really want to understand is how to harness this connectivity pattern to build biotechnologies that work under specific conditions or for specific outcomes. But this is very difficult because we're essentially dealing with uncultured communities of microorganisms, and we can really only access them through their genomic information. And so in effect, we have to convert the network cellular structure into genomic sequence information, and we have to learn how to read that matrix of ones and zeros to build those biotechnologies. And so we do that through the lens of biological information. We can look at DNA, which is the hereditary information that records the evolutionary history of the cell. We can look at that through RNA, which is a messenger, which communicates the message from the DNA to the ribosome, which builds proteins, and proteins down here do the work. So we can look at all three levels. We talk about that as multi-omics. When we sequence the microbial community as a total, plurality, we refer that as metagenomics. If we look at the RNA, we refer that to metatranscriptomics. If we look at the protein at that totality level, we call that a metaproteome. So one of the things we can do with the information is build molecular barcodes that help us differentiate between one community of microorganisms and another. And you'll hear from Rachel. Yeah.
Just quickly, we can't actually see your slides. Are you sharing?
Oh, I am. Oh, you know what? I'm so sorry. That I thought that was automatic.
No.
All right, well, let me get in here, and I think I'm sharing now.
We can see now. Yeah.
Okay, so I'm just going to pick up right here at the matrix. This is what happens when I can't see the screen fully. So I was talking about biological information, and I was talking about how microbial communities are, for the most part, the ones that we're interested in are not accessible through cultivation. So if we really want to harness the power of microbial communities to build new biotechnologies that will help build that mine of tomorrow, we're going to do that through the lens of genomics. And that involves sequencing the hereditary information of those microbial communities as a totality, as a plurality, as a community. We can also access other forms of biological information. That could be the RNA, which is the expressed information from the DNA that's used to encode proteins. The proteins are then produced at the ribosome, and those are the entities that do work inside the cells. They're the catalysts that we also want to harness. And so we can use that biological information as molecular barcodes, and you'll hear from Rachel how she's using molecular barcoding in an exploration context to look for mineral deposits. You can also use them as biomarkers or diagnostic tools for monitoring the performance of reactors or processes that you're interested in if you're remediating tailings or something like that. You can also use the biological information to reconstruct the metabolic potential of the microbial community. And this is where it gets really interesting from an engineering design perspective. So we can look at these genetic pieces of information and how they fit together to build machines, the catalysts that actually do all of the interesting work. And we can both use those directly from the communities that occur in mining sites. We can also use synthetic biology approaches to build new machines that do specifically what we want. But there are some very big challenges to navigating that matrix of information that we are able to generate now relatively easily at lower cost. And that is that we have to reduce the time to completion of research because the data sets are very large and they're very complicated. That often require many people to work through this information to extract meaning. And so we want to be able to reduce staff and expertise required to read that matrix. And of course, there's end-to-end storage, documentation, and reproducibility issues. So there needs to be a whole platform, in effect, that allows us to convert the biological information into actionable insights. And so that's really what I wanted to touch on today. One of the approaches that my group has been developing over the last almost decade is bioinformatics pipelines that help us read that biological information in the matrix. Here's a pipeline called MetaPathways that takes the sequence information and through a series of stage processing steps renders a database and a metabolic model that can be used to look for interesting catalysts within a microbial community. We call these models environmental pathway genome databases. It's publicly available software. You can find it at GitHub. And it's meant to empower people to interpret and translate the knowledge that is within their sequence information. It has an integrated user interface, and it's also compliant on high performance computing resources. And so an environmental pathway genome database, this model of metabolism that represents the microbial community, is roughly schematized here. You have the microbial community and you have the sequence information that's extracted from that community. It's organized in a hierarchy, which is the underlying database structure. And then it can be rendered in different formats, tables or visualizations. So through the graphical user interface, you might see something like this, which is a table where you have columns that represent different samples, and you have accounting. You have the total number of sequences, you have the types of pathways that are present, and you have processing steps in the background. And so the idea is to create a uniform framework for processing information that's also easy to access by the user. You can also visualize that data looking at the community as a metabolic network or a community cellular overview. You can zoom into individual reactions all the way down to the genes that encode them. And so in this way, you're able to look at the community as a cell instead of, all that complexity overwhelming you. I wanted to just briefly show you how we applied that with some data from Sue Baldwin, who also will be presenting today. As you know, there was an incident at the Mount Poly site some time ago, which resulted in spilling of tailings into the environment. And Sue's group has worked over the years to try and find ways to help remediate that. Here's a picture of that dike that broke. And so she provided samples from these biochemical reactors down here that were involved in selenium and sulfate removal, generated DNA sequence information from these different reactors. And we compared the results from those reactors to another type of bioreactor involved in enhanced biological phosphorus removal. So more on the wastewater treatment side of things. And we took all of the sequence information and we created essentially a barcode for each sample. And then we used statistical methods to visualize the relationships between those barcodes. And what you can see here is that there's a nice clean separation between the bioreactors doing the phosphorus removal and an intriguing separation between the two biochemical reactors involved in sulfate and selenium reduction. We can compare the totality of pathways by doing a type of set difference analysis and just say what types of functions are shared between those different reactors and what types of functions are unique. And that might be very important in engineering or improving a process. We might be focusing on those unique pathways that differentiate them. So there was a core of almost 1,000 different pathways that were shared. And then you can see that there were some specific pathways for the two biochemical reactors, about 58 for one and 20 for the other. And then we can zoom in and we can start to look at the differences between those pathways. So they're clustered at the top, and you can see again that those BCRs are differentiated down at the bottom here, these 4 samples, from those involved in enhanced biological phosphorus removal. And then we can specifically ask what types of pathways are unique, those ones at the edges of the Venn diagram. And then we can go in and we can see that there are pathways for phenol degradation, or thiosulfate or sulfite oxidation that are specific to those BCRs that may be relevant to the process that is being engineered. And so we can also take that a step further and apply something called biomining, which is a way of functionally validating pathways that we might identify in silico using the computer by essentially developing screening tools that go after the proteins that are being expressed. And the way we do that is we'll extract the DNA, and instead of sequencing it, we'll build what's called a library of those DNAs. We'll put them into E. coli that we can grow in the lab. And then we'll actually probe for the functions of interest, whether it's sulfur reduction or whether it's selenium detoxification. We can build a screen in the laboratory to do that. We can identify the biocatalysts, and we can ultimately use them in synthetic biology applications to engineer new strains. So the last thing I wanted to bring up was that all of this needs to be done within a knowledge-based framework. And so we would like, through BRIMM, to be building an architecture, a cyber infrastructure, if you will, that would collate a lot of the mining-related data sets into a common framework that could be used by both academic and industrial users to improve their processes. So in conclusion, I told you about the microcosmos. There are 10 to the 30 bacteria and archaea on the planet. They're organized in metabolic networks. We can develop new search functions using computers and specialized algorithms to tell us about the genetic composition and metabolic potential. And we can use various functional screening methods to actually build new types of microorganisms that ultimately allow us to build the mine of tomorrow. And with that, I will just say that there are some very exciting things that we can do going forward with the development of tools. And you're basically going to hear about this looking forward during the rest of this webinar. So I'll end on that note. Thank you very much for your attention.
Thank you very much, Steve. Really exciting. I did biochemistry back in the 1980s, and I'm still amazed at how far it's gone. It's a really advanced science now that has lots of industrial applications, so that's very exciting. We had one question, and the question was from someone in Kenya, I believe, and as she said, do you know of applications or to treat mercury in mines?
Yeah, so that's actually a very interesting question. I've collaborated with several people who are interested in methyl mercury and its fate in the environment, and we can definitely identify genes that are implicated in that process in different types of environmental samples. And so the short answer is these tools are directly applicable to that problem. Of course, it requires not only analyzing the data, but ultimately validating it. So I would just encourage people to think about that. It's one thing to go looking for the information. It's another thing to actually take that information and action, realize through actions some function. And that's the part that I find most exciting. And I think you'll hear from Scott on one of the ways that they're doing that to actually pull minerals out from complex ores.
Excellent. Thank you very much. And